So my name is Margaret Anderson, and I am the executive director of a group called Faster Cures. We are based in Washington, D.C., and we're a center of the Milken Institute, and we look exclusively at the medical research system. So I'm delighted to be here today. We have a rock star group of panelists, and I'm going to do my best to stay out of the way and let them tell us about this new world of precision medicine, custom cures. So we're going to start off with some discussion at the kind of the 30,000 foot level of what the science really looks like. If you could pull up slide 1, which is up there. I mean, you know, this is one of the images that I chose to illustrate what I think is happening out in the popular press. That I think that the American public and perhaps beyond are starting to think that, gee, you know, I'm going to be able to go to the doctor and get a pill that literally has my name on it. If you can go to slide number 2, we have Dr. Francis Collins sitting in the front row here. And Francis, you look just the same as that Time magazine cover. You know, no worse for the wear. So, you know, we're going to be talking about the Human Genome Project and what kind of fruit is being born right now. And I think this year there is more excitement and promise perhaps than ever before. But as you're going to hear from individuals like Retta Beery, who is the chief medical officer of her family, you know, we have a little bit of a ways to go in terms of how do we actually deploy the knowledge and mesh it with practice. So, as I said, if you turn to slide number 3, you're being bombarded with headlines, both in the science press, but also the popular press and mainstream media. And what you're seeing in terms of this topic of precision medicine is that the pace of interest in this area is really just accelerating. If you turn to slide number 4, my own work, my own career has always been in science policy. And the first job that I had out of grad school was to work for a congressional agency called the Office of Technology Assessment. And I put this slide up. Love the graphics, right? We've come a long way in graphic design. I think somebody, you know, literally drew those on her desk and put them on the cover. This was a series that we did for Congress way back when, and it's called New Developments in Biotechnology. And this was looking at U.S. investment in biotechnology and what we were going to sort of reap from that experience. We've come a long way, but we have a way to go. So if you could turn to slide number 5. Let's start with Mike Pollini, who is going to talk a little bit about how we got more than just the T-shirt out of the deal in terms of the Human Genome Project. And I'll have you know that I took this photograph last week at the NIH Clinical Center in the gift shop. So the T-shirt is for sale, literally. So Mike Pollini is the president and CEO of Foundation Medicine. And let me do some other quick introductions before we get going. To my left is Jim Greenwood, who's the president and CEO of the Biotechnology Industry Organization, but also was a member of Congress. And I heard you say you had public service for 24 years.
That's right.
Before your work at Bio.
That's correct.
How long were you in office?
24 years.
In Congress?
I was in Congress 12 years. I was in the state legislature for 12 before
Okay. So Jim is
I was a social worker before that.
Excellent. I'm sure you're still a social worker
The only
now
Republican
in Washington.
social worker in the history of Congress.
So Jim is going to be talking about the business model and some of the public policy issues. To my right is George Church, who is a professor of genetics at Harvard Medical School. Again, I said Retta Beery to George's right, who is a patient advocate and speaker. And you're going to be hearing a little bit about her personal experience and her passion in terms of integrating the patient voice and need. And then to my far right is Dr. Chris Austin, who's the director of the National Center for Advancing Translational Sciences. I know we keep botching that acronym pretty much at every turn.
Yeah, your boss does.
So you can turn it into anything you want, and maybe we'll get people interested in funding that center at the NIH. So, Mike, can you start with some framing sort of work for us around the science? Because we are aware that not all of you have MDs and PhDs, and some of us took biology a long time ago. So, you know, take us on a little, you know, science course so that we can sort of frame the discussion today.
So I'm out probably 3 or 4 days a week speaking about personalized medicine, where the world of targeted therapy is going, especially in cancer. but I feel like this is a bit of a setup to ask to give an introduction on the science with George Church on the panel and Francis Collins sitting in front of me is one of the most unnerving things I've ever done in my entire life. And so what I'm going to do is I'm going to start way over here and then look different directions for additional
They're
comments.
They're going to critique when you're done.
That
okay?
is, now it's even worse.
This is like Shark
It's
Tank
even,
or something.
That's what I feel like. So my kids should be watching this. They would give me a hard time. Well, I think there are a couple of examples though, that we can turn to even from this past week about some of the confusion within this space, especially as we talk about sequencing, next generation sequencing, personalized medicine. What does it all mean? Actually, both of the examples that I'm going to point to come from the New York Times within the past 7 days. It was last Monday or Tuesday where there was an article about utilizing whole genome sequencing and different types of sequencing to direct personalized therapy. Now, in that article, not to be a critic, I try to avoid saying these things at least publicly, but there was a transition, a shift back and forth between germline testing and testing in cancer. And it was clear that, I think it was relatively clear, that the author did not necessarily fully appreciate the difference, and it's a very important difference between thinking about understanding our genome in the context of germline disorders, so meaning errors of inborn metabolism, things where we're typically looking at our blood to understand the traits that we've inherited, either that predispose us to disease, that cause disease, versus using sequencing in the context of the diagnosis of cancer, which is where it's typically performed in a very different context. Again, oversimplifying. And then if we go to the New York Times yesterday, there was a brief article, again, in follow-up, and that article focused specifically on sequencing in cancer to direct targeted therapy and what are the tools that are necessary in order to direct targeted therapy against one specific cancer. It was focused, it hit the point. And so I thought I would really just throw these into two buckets and then again look to my right for additional comments. So as we think about sequencing itself, why do we care about sequencing? Well, sequencing helps us understand the nature of disease at the level of our DNA, at the level of our molecular blueprint. But again, let's think about, let's look at the spectrum of sequencing. We start at the one end, which is whole genome sequencing. That's the original, more or less the original genome that was on the cover of Nature and Science. How many years ago? 12 years ago now. And where we mapped the 3 billion base pairs to understand the full context of one's genome. Then as we migrate a little bit to the right, you hear the phrase whole exome sequencing. Whole exome sequencing, again to oversimplify, but that's really where we're not necessarily looking at the one's entire genome, but we are looking at the coding regions. We're looking at very specific regions across all the genes. And then as we continue to migrate to the right, we get into the area of targeted sequencing. Targeted sequencing means we're not looking at the whole genome, we're not looking at specific areas within all of our genes, but we are identifying a subset of genes. That might be 10 genes, it might be 500 genes, but we are identifying a subset of genes that we are electing to sequence to better understand a patient's condition. So first, there's that spectrum of sequencing that is performed today in order to help us how to treat patients at a very individualized basis, at a very individualized level. Then we'll shift over maybe a little bit more to the medical side and away from the science. I've mentioned germline testing, so I'll oversimplify, but germline testing, think about doing your sequence on a blood draw. We're looking at, for the most part, we are looking at the DNA that you were born with. We're looking at changes in one's DNA to help predict disease, to help understand diseases that you might have just been born with and are now manifesting themselves. It's actually, and so typically we often tie whole genome sequencing to blood-based or germline sequencing. Oftentimes that's how it's used. Those are the connections in the clinic. Then if we migrate over to the world of cancer, which is doing sequencing looking for somatic mutations. Well, what does that mean? Somatic mutations mean errors of our genetic code that we're not necessarily born with, but that mutated over time. Alterations that developed as we go through this aging process. Somatic changes are the changes that we are looking for to understand one's cancer. I'm going to pause there because I just wanted to hit those two buckets, and I'm going to look to my right where George can give you the real deal.
Say, please look right past me.
No, I was going to say, Jim, would you like to weigh in on that in any way? I'm kidding.
Oh, you're kidding.
George? No, I'm going to get back to you on the business end and the policy end. George, do you want to add anything to that? In terms of the understanding for the audience?
I'm certainly not going to go any deeper dive than that. I think the important thing is that for cancer, we have the opportunity of doing preventative as well as curative, and that's a very big deal. If we have a heads up from the inherited genome, then many patients elect to have prophylactic preventative surgery on a whole variety of different tissues that they're prone to, and this has been a wonderful possibility. I was just at a session on HIV, and the entire session went by without really discussing a cure for HIV that is in the form of gene therapy that Sangamo and others are testing, where you can change your genetic composition. And this is in phase 2 clinical trials. And so when we say that genetics is not your destiny, it used to mean you can change your environment, surgery, drugs. It now means you can change your genetics as well. And I think that's an important thing that should be out in all these conversations.
Okay. So, Retta, I would like to turn to you and ask if you could share your story from your family's perspective, because I think this story will really punctuate the point of some of the issues that we've already started to talk about and that we'll continue to.
Sure. So my husband and I have 3 children, and 2 of our kids, our twins, Noah and Alexis, were born with some medical issues. At 9 months of age, we took them to their pediatrician. We were sent into a world of specialists and doctors and testing and a lot of unknowns. So when they were 2 years old, we had an MRI done, found brain damage, diagnosed with cerebral palsy, and then at age 5.5, our daughter had started to get worse. She started to lose a lot of her ability to function. She was unable to walk for the majority of the day. And do you actually have my slides in there? In the first 5.5 years of their life, I've got a slide that represents some of the tests and the specialists that we took Noah and Alexis to. I think, could you go back one? Right there. So on the left-hand side of the slide are some of the specialists and the doctors that we took our twins to in the first 5.5 years of their lives. And on the right-hand side are the results from all of those appointments. A lot of invasive testing, a lot of hospital visits, and the results were an inaccurate diagnosis, and a lot of physical and financial and emotional costs were involved. I found out I had done research for about 4 years. I found an article that told me of a different diagnosis that my daughter fit the symptoms of. And so we got an initial diagnosis through an article that I found. We started our daughter on a medication based on this article, the drug L-Dopa that they give to Parkinson's patients. And she went from wheelchair-bound, and we were talking about feeding tubes, to being able to function and do things that we never thought were possible. Ultimately, we ended up starting my son on the drug as well, and he had the same response. So fast forward to 2008, my husband took a job at a company that was in Vitro. They were getting ready to acquire Applied Bio. They made my husband the offer based on the promise of this technology, this whole genome sequencing that they were going to start doing. And it lined up with some of the things that we were doing with our kids. So for years, in 2003, I'd started a website, and for years we'd reached people around the world that had been misdiagnosed, and they would ask me how they could find out if their child or themselves have this diagnosis that our kids have. And it was always a response to a medication. So the idea of a diagnostic tool was fantastic. We moved to California in 2008. And then in 2009, a year later, our daughter, who had been struggling with a cough for many years, it turned into a severe, life-threatening breathing problem. We almost lost her a couple of times with paramedics coming into our house trying to get her to breathe again. So we went through, if you could, oh, there's the slide. For 18 months, on the left-hand side of the slide, we went to, again, a myriad of specialists and tests and invasive procedures for my daughter, only to have no answers. And in the meantime, she went from running track and playing basketball to not being able to walk down the street without needing a breathing treatment. So in 2010, we engaged in a project with Life Technologies and Baylor College of Medicine, the Human Genome Center, and we decided to have Noah and Alexis's whole genome sequenced. So when they had, if you could move to the next slide. Oh, I'm sorry, one back. You know what, we'll stay there. It must be at the very end. So, so we had Noah and Alexis's whole genome sequenced. We found the genetic mutation responsible for their neurologic disorder, and it told us that not only were their dopamine levels low, which is what we had been treating for the last several years, but their serotonin levels were low as well. And so now you can go to the moving slide if you would. So this was Alexis before we discovered, this is actually her right at about age 5.5. The moving video.
We'll get there.
And so Alexis and Noah, now, for the first time in their lives, we had a definitive black and white answer for their neurologic disorder. And we had evidence that gave us a correct diagnosis, and we also had, that's it, perfect. So this was Alexis before. This was her at about age 5. And as you can see, she had absolutely no control of her body. She had an inability to even walk by 10:30, 11 o'clock in the morning. She couldn't sit up. And then if you could go to the next moving video, the one right after that. Alexis went from that to being able, this is after whole genome sequencing and adding the additional
Good job,
therapy.
Alexis.
So she runs track. She has broken
what
track
the final
records
results
as a freshman. She's very excited. She's very enthusiastic. So this is what we had for whole genome sequencing. And then the very last slide, if you don't mind, is actually what we went through for whole genome sequencing. We had to have their blood drawn. We signed consent forms, and the results on the right-hand side were an answer and a new life for our family.
So thank you for sharing that. I think it really illustrates the, sort of the power and the promise, but also in a way some of the despair in terms of really trying to get to that answer. I mean, here's the slide that you had referenced. So Chris, I'd love if you could comment on this. I know when we were discussing this panel, you were bringing up some of the relevance of Retta's family story to some of the broader work that you're doing around rare diseases. And what are your observations from this? I know you're familiar with the story.
Yeah, so I think this illustrates the promise, but also the peril that we're in. That in the last 10 years, the ability to do the kind of thing that you just heard about has become possible. But the medical system that we have is not suited to do this in an efficient way. That's point one. Point two is that this is a wonderful story because there was a treatment available once the diagnosis was finally made. Numbers to keep in your head is that there are about 7,000 diseases just like the one that you heard about, 6,500 of which have no treatment. So even when one has a diagnosis, there's no therapeutic implication of that. And so the bittersweet part of this is that we are, as a community, becoming very good at doing sequencing. And I'm a genetics guy, like most of us, so that is my first love. But I must say that the difficult part now, from my point of view, is not the sequencing. It's actually how to interpret the sequencing, how to match it with the clinical part. We always talk about genotype, phenotype correlations, if you've ever heard that term. What that means is you got a mutation or a change in the DNA sequence, and how does that manifest in a person? What kind of disease or what kind of change, what kind of trait does it produce? We have become very good at doing the genotype part, and to a great degree we've forgotten about how to do the phenotype part. And so one of the things I think we have to do to bring the promise of genetics to patients is to focus more on that phenotype part, the clinical manifestations part, because you just heard that was the problem. This diagnostic odyssey that you heard about is absolutely typical, 2, 3, 4 years. I think the other difficulty that those of us in this field are dealing with is that though there is enormous promise, an enormous amount of knowledge, and just throw another number at you, in the last about 15 years since the large amounts of sequence data started going into public databases as part of the Genome Project, about 4,500 diseases have had their molecular genetic basis discovered. And so that shows you how powerful that knowledge is at producing diagnosis. But what we ultimately want, and I don't want to minimize diagnosis, because for patients with a mystery disease, having a diagnosis is really important. But of course, ultimately, what patients want, what all of us want when we have illnesses, is an intervention. And unfortunately, the diagnosis it does not lead directly to a treatment in any direct way. Those of us who are geneticists often get, who talk about the power of genetics for therapeutics, often have hung in front of us the specter of the first disease, the molecular basis of which was discovered, which was sickle cell disease in 1949. There is still not a drug for sickle cell disease based on that molecular discovery. So it is not for want of trying. And so that's what the center that I run at NIH, this new NCAT center, is trying to change. Because it's very clear to me and most of us that the system we have is very good at doing what it was supposed to do over the last 50 or 60 years, which is to create understanding of what causes disease. But it's really poor at translating those discoveries into interventions which tangibly improve human health. And there's a lot of ways we could talk about how to do that. One of the ways is exactly what you just heard about, that there are about 2,500 drugs that have been approved by FDA, a few more, another 400 or 500 if you include regulatory agencies from around the world. You probably all know, from examples you may have heard of, that drugs that are useful for one thing can be used off-label for another, and very often it's very hard to predict when that will happen, because drugs, and we all know this too, drugs tend to have effects other than what they're intended to have. And sometimes those effects are bad, but sometimes those effects are good. And so when you get a diagnosis of a rare disease like this through sequencing, it doesn't necessarily mean that you've got to go through what's currently a 10 to 15 year billion-dollar odyssey to create a drug. Sometimes you can so-called repurpose a drug which is already on the market for that rare disorder.
So George, thank you, Chris. George, talk to us a little bit about the perspective that you bring to this and the work that you've done, particularly the Personal Genome Project. How does that relate to some of the issues that we've put on the table?
Well, I think the Beery family is a perfect example of how critical it is to have participatory medicine, and I think that that's a blind spot that we've had. Chris also mentions that we haven't been very good at collecting phenotypes, and I would add to that environmental components. And so we've been advocating since 2003 a project now called the Personal Genome Project, which tries to gather together genomes, environments, and trait data and make it shareable. That's another thing that's been missing, is that there's a lot of siloing of data. Part of it having to do with privacy concerns, but clearly there are alternatives to this. When the Million Veterans Project asked the potential volunteers if they wanted to learn about their own genome, as happened in this wonderful family, 96% of them said that they did want to know about it. If there was something that, whether or not there could be something that helpful for them, but certainly if there's something that could be helpful for them. So far, there are a very limited number of projects that have figured out how to do that. Personal Genome Project is one of the ones where we do give the data back to the individuals and help them learn how to do it. Now, you could say they're not educated enough. You could say the same thing about voting. We can prevent certain people from voting because they're not educated enough. I think the solution is not preventing them from voting, but giving them a vote, and then everybody becomes highly motivated to get them educated. And I think that's what we really need here, is fewer silos, participatory medicine, environmental trait data that's shareable worldwide, because there are people out there of all ages and all different disciplines that can have really out-of-the-box contributions to this sort of thing.
So talk a little bit about the environmental trait part of
Yeah.
this equation. What are you looking at there?
Well, so for example, the microbial components are increasingly being found and experimentally proven to be causative, where you can transfer obesity from one rodent to another by changing their microbial community. There are microorganisms that cause neurological problems, both in humans and in animals, such as toxoplasmosis. So we're just beginning to touch on that. There are many microbial causes for cancers, ranging from stomach cancer to cervical cancer and so forth. So that's an important environmental component. And interestingly, we brought down the price of inherited genomics by 1 millionfold. That also applies to microbial genomics, and it applies to our immune response to it. So the immune response can also reflect non-microbes like allergens and so on.
So if someone wants to participate in your work, what do they need to do? And I think we have a screenshot of a website. So if you could pull up the Personal Genome Project slide for us, what do they do?
Yeah. So
Slide 7.
It's been up already. Personalgenomes.org. You can go there. You can learn more about it. Decide whether it's of interest to you or to someone that you know. It's certainly not for everyone. We don't need to have everybody on the planet right now. In fact, couldn't even afford it if we did. But it's an opportunity to share in the same way that this wonderful family has, to go out there and say, this has been our experience. We need precision medicine, not just whole genome, but all these other things, environments and traits. We have a large data set which we can share about an individual, not something that's where we take a little sliver out and share that in a very limited way. We need something that's really large and shareable.
Okay.
So if anybody's interested.
I want to, we'll keep coming back to this topic of open data and how scientists can access that and how it's going to advance things. Jim, I wanted to turn to you. You have been heading up the bio organization for some time now, and so you've really seen this evolution of the science and the business model, and even the evolution of the terminology that we're using from personalized medicine, which I fear might have made people think they were going to get a pill with their name on it, to precision medicine. You also just convened 20,000 plus people for the bio convention.
Actually, I wish about 13,000.
Okay, 13,000 people in Chicago. Tell us what the business model is for this, and maybe what some of the opportunities and potential challenges are.
Sure. So when I think about personalized medicine or precision medicine, I think about the right drug to the right patient and the right dose. And that's what a lot of this is all about. So we've all watched television and seen the drug ads, and while the children and folks are running through the fields of flowers, they're saying this, some people, this causes your ear to bleed and your eyes to go blind and your heart to fail and so forth. And the reason for that, of course, is, I mean, if it had that effect on everyone, we wouldn't get the drug approved and market it. But it does have that effect on a very small percentage of people, and as a society, we make the decision that and the patients do, that the risk is, the benefit is worth the risk. But the ability to figure out, first off, which people should not take that drug. So there are some people who have those side effects, and in not all instances is it genetics, but in many cases it is their genetics that makes them not a good candidate for that drug. So being able to identify that, and as George said, not just on the genetic sequencing factors, but other factors about the personal medical history of that individual, says this drug is not for you. And that can in itself avoid obviously a lot of disability and death and expense of hospitalizations, unnecessary hospitalizations. Then the thing is that we want to find out that we understand that there are certain drugs that are only for certain people. So, starting with Herceptin, that treatment for cancer is only for women with certain genetic qualities. There are people who have, for instance, a mutated KRAS gene that says for them Erbitux works for colon cancer, but it doesn't work for others. And there's a new drug called Xalkori, which is for non-small lung cancer where the ALK gene is mutated. And so those drugs are precisely for those people and for no one else. And by understanding the genetic predisposition to either respond well, not at all, or adversely to the drug, we obviously do more harm than we do good, and we save a whole lot of resources at the same time, because we're not dosing people who don't need the drug or at all, who wouldn't respond to it. So I think that's very much the promise of the drug. We're also able now to understand how people have difference in their genetics that determines the ability for them to process, if you will, or metabolize a drug. So if you look at warfarin, for instance, the blood thinning drug, that has to be given in just the right dose, or it can actually, if it's improperly dosed, you're going to have people getting strokes anyway. And so understanding the genetic differences in people in the way they metabolize drugs enables the physician to more precisely dose the warfarin, and we think that already we're saving about 17,000 strokes a year because of that capacity. So I think, whereas it's true that for the large percentage of diseases we don't yet know the genetic cause, we don't have a therapy, we're certainly moving in that direction. We're moving in the direction of being able to identify the genetic cause of a disease and then develop the therapy based on the genetics. And what you're increasingly seeing is what we call companion diagnostics. So the FDA is saying you develop a diagnosis that determines for which patients this drug is applicable, and then develop the therapeutic. And when you market these things, the therapeutic will say only to be used after the diagnostic test has been performed and deemed this patient to be appropriate.
So, Mike, you helped us out in the beginning with the science, but talk about the model at Foundation Medicine and what you're doing and how it relates to some of the things that you've already been hearing about.
So George mentioned Magic's discussion earlier today, and HIV is a wonderful model as to how we think about what's going to happen in cancer. It's starting to happen today, and clearly we are at the tip of the iceberg here. And so what does that mean? It means that we're going to take a disease that in many instances is a death sentence, and over time, thanks to the diagnostic approaches, thanks to the therapeutic options, it is going to be turned, many cancers are going to be turned into chronic diseases. Before we think about cure, although a lot of people are thinking about cure, the idea is to go through this transition from a death sentence to a chronic disease, just staying ahead enough of everything so that next therapeutic, as you heard, will be available for the patient. Our company chose to focus on cancer, right? This is an enormous space. Cancer it's not a disease. It's a set of thousands and thousands of diseases. So why did we choose to focus in that specific area? Not just because it was the hardest, but because the clinical applications within cancer are perhaps the most obvious, the most transparent. We're still dependent on the therapeutic companies. We can, in fact, do much of the diagnostic analysis, much of the genomic assessment of a patient's cancer today. The challenge is just because you find something doesn't mean that you can actually do something about it. And so that's also what we're focused on, is not just finding information, that's left for the R&D world, but finding information that a physician can act upon. And what does that mean? While there are other areas that we'll touch on outside of therapeutics, it means that there's a drug on the market, or there's a drug in clinical trials, or within a year or two, we think a compound will enter man, so there will be another clinical trial that can in fact impact these changes that we're identifying. And so if we look at, Jim just mentioned the notion of companion diagnostics. So today, what happens when a patient is diagnosed with cancer, as unfortunately many of us know in this room? Individual tests are selected, and we try to understand that patient's cancer, right? We shine a laser beam here, we shine a laser beam there on the tissue, and from a couple of quick data points, we try to understand the cancer. Well, I think what's important to understand, and fortunately most of the oncology community understands, is that we can no longer do that. Cancer is not a disease of a specific tissue type, of a breast cancer, of a colon cancer, of a lung cancer, of a cancer of the gallbladder. We now know that certain molecular pathways are activated and inactivated, and for the most part, they don't necessarily know that they're residing in the breast, that they're residing in the lung, that they're residing in the prostate or elsewhere. We're really overlapping. And so what that means is that the complexity of these diseases is tremendous. And we can no longer shine a specific laser beam in terms of a test, a companion diagnostic test, or 5 companion diagnostic tests, especially when the tools exist where we can now shine the floodlight on each patient's cancer and let the cancer tell us what the key molecular drivers are. Once we do that, we can then marry this information up to the therapeutic options. So it's fundamentally a different way of understanding how we think about diagnosing and treating a patient with cancer. If we stay with a paradigm where the paradigm in which we've lived over the past few decades, we are going to continue to hit major roadblocks. What are those roadblocks? First of all, biopsy techniques are improving. We are running out of tissue. You run a handful of individual molecular tests, there's a good chance you won't have enough tissue to run the next handful. So we're wasting tissue. Second of all, these costs are escalating. Every time you run a single molecular test, on average, you're thinking about approximately $500 per test. Start to run 8, 9, 10 tests, $4,000, $5,000, $8,000, $10,000 in non-small cell lung cancer if you're going to an academic medical institution for an assessment. The payers just aren't going to put up with that. They don't have to put up with that. The world's changing. We can think about new ways. And the other theme that's really emerged is this notion of, all right, we are getting flooded with information. What do we do? How do we assist both oncologists and patients and other caregivers in really working through all this information? What we do at Foundation Medicine, aside from doing the genomic characterization, that's about, you think, we're in Cambridge, Massachusetts, it's the biotech mecca, it's all technology. That's part of the equation. 50% of the equation is how do you take all this information that's generated and translate it so it can be seamlessly integrated into the practice of medicine. If you give an oncologist a CD with genomic information and say, figure out what to do with it, you're dead in the water. Nothing will be done with it. So what we have to do is focus as much on the IT component. It's why Bill Gates is an investor in our company. It's why Google is an investor in our company. It's why Yuri Milner on the tech side is an investor in our company. Because we spend half of the time that we spend, half of our investment is in thinking about that second half. What do you do with all the information all over the world? How do you marry that up with a patient's cancer and help that oncologist and the patient make the best treatment options for his or her specific disease? That's the whole spectrum of challenges.
So I think you just cracked it wide open, I think, for the audience, which is that we're talking about this incredible, exciting science and promise and data and business opportunity and patient need, but we have a system that is it really ready to accommodate all of this, and what does the system need to do to be ready for it? Retta, I'll ask you to talk because you've been living this, but also working with other sort of patients and families. what's your advice to people about the role that the patients, are going to play in this giant stew?
I think that patient advocacy is a must. I think that we need to demand the diagnostic tools that are available, that are becoming available. I work with patients and parents of patients that have exhausted every test that the clinicians know of. And I believe that whole genome sequencing, especially when you're dealing with someone that is undiagnosed, something that is rare, something that they can't identify through symptoms, I think that we should start with whole genome sequencing and then go from there. I think the slides illustrate the cost of going the traditional route. There's a checklist that patients go through, that doctors go through, and they start with blood tests, they go to MRIs, they go to spinal taps. I think we should start with whole genome sequencing.
Other comments from panelists?
Well, just the only thing, I think it's the American College of Genetics and Genomics that just came out with a recommendation for which are sort of the top tier. I think there were something like 57 of these tests that they thought ought to be run. I don't know whether they meant sort of, if you're going to do sequencing, these would be the places you would look at first. Do I have that right, guys?
Yeah.
Yeah, there was just a
Yeah,
report
they weren't
that was
necessarily
released on
focused on cancer, though. They were focused
Right.
on other conditions.
Right.
But
So, I mean, I keep putting myself in the shoes of whether it's your family, Retta, you basically had to gather all of this information and sort of do it yourself. How are we going to get to a place, and Michael, maybe this is a question for you, George, Chris, weigh in, Jim too. Okay, so are we asking every single family out there to sort of fight it on their own? And how do we intersect with this medical community? I mean, you talked about, what if you bring this data to an average oncologist, what's going to happen with it? And how fast can we expect this paradigm to be shifting? Because I don't want to leave people thinking that, okay, this system is a big mess and, we're never going to get out of it, because what you're hearing about here on this panel is really revolutionary, and it's starting to accelerate. So how are we going to bring those systems together, the sort of medical practice piece with the research piece? Is it happening in real time?
I think it is. I mean, I think this family is not going to be considered exceptional very soon. When we panic about big data, just think about what your cell phone does in terms of navigating the streets. When you have really good big data, like GPS data, rather than just merely map data, suddenly it's a game changer. And that's what we have now, is that patients and oncologists and general practitioners won't be looking at mountains of data. They'll be the way you look at your phone. It doesn't give you all the coordinates, the satellites and the delay times, and it just says turn left. And that's what's going to happen, is we need to vet that. And as a community, like this report that Jim just mentioned, we need to reach consensus on where you draw the line and say, below this, you're entitled to have that data, but just put it in a vault or play with it. But above the line is where it's medically actionable, and we believe as a community it's valuable to you.
Now,
and just forget about everything that it can't do, because every medical diagnostic has something you can't do.
Now, you brought up privacy issues. Talk about what are the privacy issues that are out there being debated about this?
Well, there's kind of two classes. One is privacy for the patient, and the other is privacy for the research subject. Okay, these are very different worlds. But they have something in common, which is the technology is changing so quickly that in principle, when I shake your hand, I have your DNA. Or maybe just it's all over me, even if I don't touch you. So, and to some extent, it's my right. It's now my property. It's on my skin. Do I have software that's going to prevent me?
You can have it. It's okay.
Yeah, right. I mean, not everybody is quite so open as you are, but but I mean, this is a new challenge, right? So, I mean, is our DNA going to be like our faces? Our faces not only reveal our ancestry and our predispositions, they tell us what diseases we actually have and whether we're emotionally upset or calm or whatever.
And
so
my lifeguarding career right here, right? In terms of my brown spots.
Yeah,
there you
absolutely,
go.
yeah.
Sun exposure.
So I think that for some people, the genomes will be like their faces. It'll be something where they feel not only it's safe to share, maybe it's not totally safe, but maybe you get benefits that outweigh the risks, and maybe there's some obligation there.
Do you think that, I want to just talk about this privacy issue for a minute. Do you think that people's perception of privacy changes when they themselves become patients or people in their life? become patients, in that they're a little bit more interested in open sourcing data to accelerate things? I mean, was privacy a top concern for you, Retta, as you were trying to figure out solutions for your children?
Absolutely not. And I actually had a conference call with Richard Gibbs and some of the team members at the Human Genome Center when they found the data. And they were going to write up a paper, and they were going to put subject A and subject B. And I said, don't do that. I said, use their names, because otherwise it's going to just be another paper, and it's not going to bring life, and it's not going to be something that people can connect to. And so privacy was not a concern at all. And I actually spoke at President Obama put together a commission for the study of bioethics. And one of the commission members asked me the question, are you concerned about your kids getting insurance in the future? You've gone so public with their neurologic disorder. And I said, you know, in this hand, I have the concern of insurance issues in the future. And on this hand, I have their life. There's no balance to that question at all. So I'm not concerned about privacy issues. I'm obviously I'm not concerned about privacy issues. And I think that patients want to start sharing information, because that's where you find answers. And the more we continue to hold back and keep everything private, the less we're able to do that.
I think there will always be concerns, legitimate concerns about privacy. But a couple of things that we need to do in order to make people a little bit more comfortable with all of this, and that is make sure that the stigmatization and the discrimination from particular unique genetic factors are that we work on those. So the fact that people worried about whether you just mentioned insurance. The Congress did pass the Genetics Information Non-Discrimination Act, GINA, which basically says that insurance companies and employers can't discriminate based on genetic information. Here in California, Governor Jerry Brown, I think, just passed a more recent law that applied that broadly, even more broadly, to things like housing, public accommodation, and so forth. So we have to make sure that people can't be discriminated against on the basis of this, and by continuing to destigmatize diseases, whether it's HIV/AIDS or depression or what have you. I think we also can make people feel a little bit more comfortable with this information being not in the public domain, but used for research.
So I want to ask about we brought up insurance in terms of the discrimination and the privacy angle, but I want to bring up the issue of reimbursement. It was touched on a little bit. How is the payer community being brought into this conversation in terms of understanding where the science is today, where it's going, what the patient need is? I mean, Mike, in terms of your work, and Jim, your member companies, how are they working with that payment system?
We have until 3 p.m. for this panel discussion, right?
Right. I mean, we could spend a whole week on this topic alone.
It's interesting, the transition from privacy to reimbursement. Intuitively, one might think that privacy is kind of up there on the same level in terms of the challenges as reimbursement. In our world, they're on two completely different levels. Privacy just doesn't, it's an issue that doesn't come up that often. If you stay HIPAA compliant, you do this in a thoughtful fashion, it's just you have to answer the questions, but it's not an issue in the context of reimbursement. Reimbursement is a bear. Reimbursement is something that we all need to think through as we go through the work to really personalize therapy, whether it's in cancer or outside of cancer. Now, fortunately, what we've seen from the payers is that they do in fact understand that the world is changing. So I think if we were engaged in this conversation 5 years ago, the payers would perhaps even be a little bit suspicious that personalized medicine is going to happen, the world of targeted sequencing and whole genome sequencing and doing this type of assessment for patients with disease is inevitable. They understand it is inevitable. They really do. I don't spend a minute with a payer, or whether it's a private payer or with CMS, where we have to convince them that the train has left the station and this is where we're going. And fortunately, they all appreciate that long-term it's a good thing. 5 years from now, you can pick it, I don't want to debate over 3 years, 5 years, 7 years, but 5 years from now, They understand that what we are collectively doing could have a very positive impact not only on medicine, but a positive impact on the costs associated with the diagnostic workup and the therapeutic selection of patients, again, with cancer. So it's more of a question of how do you get from here to there? That's where we are today, is really talking about, okay, should the cost, because the cost of maybe sequencing a gene has been X amount, how do we think about sequencing for 50 genes, 500 genes, or doing the whole genome? Does that mean it's logarithmically more expensive? No, it can't be. And so we have to put the onus on us to make sure that we're being smart with the pricing decisions in terms of both I'll just say on the diagnostics, because that's the area that we know. They also want to understand, how's this going to play out? Okay, we launched our first test a year ago. We've literally run thousands and thousands of clinical specimens. We have over 1,000 oncologists and at virtually every major cancer center in the United States that routinely works with our company. We've had oncologists in 22 different countries order our test. We don't have an outside sales force. There is a pull in the marketplace. So what are the payers concerned about? That this is going to overwhelm them. It's going to hit overnight. Well, we can't let it hit overnight. The world's not ready for that. And so then the discussion is, okay, how do we think about pricing, and how do we think about the types of patients that are going to benefit from this approach over time? We might want to think about certain patients with perhaps late-stage disease today. Maybe that's an easier one for a payer to bite off. We might want to think about patients with uncommon disorders or uncommon cancers, because there is no great diagnostic testing. There are no great therapeutic options for many of those patients. And so that's another finite segment. And over time, one can see it going from certain patient populations to broader and broader and broader populations. So maybe by the time we get 5 years out, every patient in the United States newly diagnosed with anything but the most routine cancer will in fact have that genome sequence, both their own genome sequence as well as, in essence, the cancer's genome sequence. It's going to play out over time, but I think a lot of the onus is on us to make sure that we work with the payers, think through this smartly, run the clinical trials, and don't force it. We have to let it play out over the course of months and years.
I think both the public and the private players suffer from short-term thinking that they're forced to for structural reasons. So the federal government, the Congress, if you said to the Congress, if you spent $100 today, 20 years from now, you'll be able to prevent anyone from having Alzheimer's disease. The Congress and the Congressional Budget Office says, well, that's going to cost us $100. And if you say, no, but it'll save us hundreds of millions of dollars, billions of dollars. I said, but we do our budgets in 10-year periods. So Congress is in a vicious cycle where it knows that the cost of health care, particularly the Medicare program with the silver tsunami of the baby boom generation, 10,000 baby boomers a day going to Medicare. They see that, it scares them to death in terms of the cost of Medicare. But they have a hard time focusing on, well, let's just put more money into treatment and diagnosis because we'll have fewer sick people, because it doesn't score in the 10-year frame that they have to do their budgets in. So that's a problem. I think private insurers have an even worse problem. People change jobs so frequently that it really is not very much in the interest of any private insurer to make an investment and long-term investment in anyone's well-being, because they're moving to another employer. And eventually they're going to move into Medicare. So why does an insurance company want to pay for an expensive test that could lead to an expensive therapy on a 64-year-old who's out the door into the Medicare, and the money's not saving them anything? So trying to figure out how to get public and private payers, we have to radically change the healthcare system. We all know this. Radically change reimbursement schemes so that it makes more sense to invest in long-term wellness for both the public and private player. And we're a long way, we talk about it all the time, but we're a long way from having our systems respond that way.
So, this is one of these panel discussions that you could have for a long time. I think there's no end to the opportunity of interesting topics, but we're going to wrap up so that we can get you guys out to your next event. Chris, I'm going to start all the way at the end and ask you if you can give a couple of key points that you think the audience needs to take away from this conversation, this topic, the work that you're pioneering at NCATS, and we'll go down the line and ask each panelist to speak for a minute.
A couple of points. First is that to solve this problem, we need to involve the patients in the way that any other consumer is involved in the development of a consumer good, which after all is what drugs are. And to me, that will, if we can get that to happen, we will dramatically change and speed up the adoption and the appropriate adoption of these kinds of technologies. Second, the opportunity is absolutely huge, both for science, for medicine and public health, and for economic development. You can think about every one of those diseases which is untreatable as an economic opportunity if we can de-risk these projects enough that one would be able to make a business case to adopt them. So we're spending a lot of time at NCATS working on how to involve patients in every project that is done, how to change the ecosystem so that is just part and parcel of any project that's done. And secondly, to work on the pressure points on particularly therapeutic development to improve the speed and accuracy and failure rate of therapeutic development. It's an unfortunate fact that whether one is developing a drug for 10 million patients or 10 patients, for the most part, the costs are the same. And as a result, that's why you see rare disease drugs costing 1,000 times as much as drugs for more common diseases. If you're going to treat 1,000 times fewer people, you just charge 1,000 times as much. As we develop more and more treatments, whether it's for cancer or other kinds of rare diseases, and I should say that all diseases are becoming rare diseases as time goes on, as we discover the basis of more diseases that we know are rare, under 200,000 in this country, that's the definition. Or in the cancer case, we're taking common diseases, lung cancer, subdividing them into less common subsets. But as we do that, the payers are very quickly, I think, going to cry uncle, that we simply can't go forward saying that, oh, it's okay, we'll develop another drug that costs $200,000, $300,000, $400,000 a year. It's just not tenable. And the only way to solve that problem is to understand the science and the operations of how to go from a discovery, a diagnosis, into a treatment. And the last thing I'll just tell you is, those of you who know me know I always talk in football analogies, so I'll give you one here. The diagnostics, from my point of view, it is a kickoff. And it's sometimes you know that there's a return all the way, there's immediate clinical application, and you just saw one. For the most part, however, a diagnostic gets you to what, the 10-yard line or the 20-yard line. Our system does not have a playbook to get from the 20-yard line to the other end of the field. It's just for a variety of reasons we don't have time to go into. It's a solvable problem. The science is there, the operation, we know how to do it, but it's going to require, I hate to say this, but it's absolutely true, it's going to require some disruptive iteration. It's going to require some creative destruction, if you want to think of it that way, particularly in an environment of at least flat budgets in the public and the private sector. We can't keep doing all the old stupid things we're currently doing and do the better thing at the same time. It's just not an option. And that is something that all of you need to join the discussion to have that happen, because the inertia to keep doing the old ways, and the people who benefit from doing it the old ways, we're all familiar with this in every area of human life, are significant. So I would just leave you with the thought that this is doable, we can do it, we know how to do it, but it's going to require an act of will on all of our parts.
So Retta, you embody the spirit of getting the job done. What advice do you have for the audience in terms of their engagement, their involvement, understanding in this topic we've just spent some time discussing?
We're all going to be patients at some point, so you have to get involved, and you have to know what's going on. You have to ask the right questions, and I think that we have to have the funding around sequencing and personalized medicine, because that's what we need. That's what each one of us needs. When they say personalized medicine, it's for the individual. It's based on not just symptoms, but exact diagnosis. And so I would just really encourage everyone to become an advocate for yourself, for your health care.
George?
Yeah, I fully agree with both of those comments. I do think we might have a playbook going forward, which is that there is one thing where I think a lot of this discussion in this room and in many other rooms is all focused on small molecule drugs. And I just think that that's a failing system. I'm just going to come out and say it, okay? I think it's the big gorilla in the room, and we're not saying it, and we're just saying, well, let's get personalized about these small molecule drugs. I say, fine, but let's start thinking about genetic therapy.
Sure.
And genetic therapy is different in two ways. One is that it's no longer about putting in a new gene in a random place, which can be destructive, but it's being able to put it in a precise place or even removing material from a precise place. That's a game changer. And it's not just happening in blood cells. It may even treat sickle cell. It can happen in, there's gene therapy in neurons in retina. There's muscle cell-based gene therapy. This is a game changer, and we need to talk about it more. We don't necessarily need gigantic amounts of fund because it is so disruptive in a good way. It could really take off. And a similar, Francis started in 2004 a sequence technology project, which was only $18 million a year, but it resulted in a million-fold decrease in the costs of the diagnostics we've been talking about here, not just for inherited. So I think a similar thing is going to happen with these advanced ways of getting gene therapy and microbial therapy, and I think we need to talk about that more.
But I would say, George is absolutely right, and you notice I didn't say the word small molecule ever. I think about these are drugs, and agnostic as to the.
But the impression is left if we don't say it explicitly.
But we are dealing. Many of the same issues still apply, and I'll just give you an example. There's a company called Provenge, and there's another not Provenge, Prosensa, and there's another company called Sarepta, both of which are developing truly personalized therapy for boys with Duchenne muscular dystrophy. And in both of those cases, they're so-called oligo therapies, or morpholino oligos for the cognoscenti in the room. But the issue is that FDA currently considers every one of those different treatments, and there's about 500 different mutations which can cause Duchenne's. And every one of those, you have to have a nucleic acid treatment has to be personalized to the mutation that you have. It's like going to a tailor and getting your pants tailored. But the problem is that FDA considers every one of those different oligos is a different drug.
So I'm going
and it has to go through the whole process.
Before we go into the FDA conversation, let me turn over to Jim very quickly.
I'll be real
and
quick, because I
Mike.
just saw the session over sign. So my comment would be, the science is galloping along. We're very blessed by that. The work that the NIH does, the work that all my companies do, is extraordinarily optimistic and promising. We're way down the road on participatory medicine, which is fabulous. Each person going online, figuring out, doing the research, taking that research to their physicians, all of the medical apps that are going to be on our iPhones, that's all going fine. What I think, we've heard a lot of concern about reimbursement. I think it's real. I think we're going to have participatory reimbursement. We can't consume all the medicine that somebody else will pay for, all the health care that somebody else will pay for. We're going to have to, if we're going to solve the problem that Congress has, and that I described, and solve the problem that the individual payers have, we're going to have to be more willing to assume more of control of our own management of healthcare expenditures, whether it's health savings accounts, or whether it's premium support, or whatever it is. We're going to have more financial skin in the game if all of this is we're going to be able to benefit from all of this.
Thank you. Mike?
So 2 quick thoughts, just to preface those thoughts. It is not a question of if. It is absolutely not a question of if this is going to occur. It's happening today. It's going to continue to happen. There are no two ways about it. Next point is, we work with many of the world's pharmas and biopharmaceutical companies as they develop their targeted therapeutics. We're involved in dozens and dozens and dozens of clinical trials today. A year ago or 2 years ago, if you were newly diagnosed with cancer, you had a friend or a colleague and you reached out to one of us on the panel, perhaps a pharmaceutical executive, and said, can you help? They would start to think about an expert in an area. They would think about a trial or 2 or 3 that maybe their company is sponsoring. Oftentimes the answer today is, first, send your, I'll be biased to plug for a second, get the tumor sent to Foundation Medicine for analysis, get it sequenced. That is now the first step. Get it sequenced, and then come back to me, the executive, the KOL, and then we can think through where to go next. That's important. The final point is, 14 months ago, based on the very first patient sample that we ran in our company, we identified a gene fusion which led to, then in our follow-up work with the Dana-Farber, it led to work that resulted in an article in Nature Medicine 14 months ago. Last month, in another leading publication called Cancer Discovery, the results of the first 3 patients enrolled in a clinical trial based on that work, they were enrolled at Memorial Sloan Kettering, were reported out. We went from, now it was a drug that was already approved in another area or was in development in another area. 14 months ago, we did not know that this gene fusion existed in these patients with non-small cell lung cancer. 1 month ago, it was reported out, and the response rate for the first 3 patients was 100%. So we went 3 for 3. It shows you how quickly this can work if we do it well, and if the companies and the academic centers and patient advocacy groups and patients, et cetera, and the payers are all working together. It can happen and it will happen.
So we hope that we've given you a sense of optimism about the future and a fast and furious look at a few of the challenges, and really invite all of you to engage with our panelists in this effort with Faster Cures, and stay tuned for the future as we update it. Thank you very much. Thanks.